So today, I'll be talking about oxidative stress. You've already mentioned that, so I'll just start from scratch. So neurodegenerative disorders is the group of slowly progressing, inherited or acquired sporadic nerve disorders. And they are characterized by progressive death of nerve cells, and neurological symptoms are dependent on the particular area where this neurodegeneration is taking place. Sporadic forms account for 95% of the whole number of neurodegenerative pathologies, and they are the widespread causes of disability and death in the world. By different judgments, the number of people with neurodegenerative disorders will be increasing and will be doubling every 20 years. And despite the decades of fundamental and clinical trials, most, if not all, the neurodegenerative diseases are poorly studied and are not efficiently cured. In our work, we focused on studying the acquired epilepsy and Alzheimer's disease. And we are proceeding from the fact, presuming that these progressing disorders have the same paths at the initial stage of these pathologies. And in addition to that, convulsions is the widespread comorbidity of the Alzheimer's disease. And almost all the neurodegenerative diseases have common risk factors. So some of them you can see on this slide. Those are cardiovascular diseases, traumatic brain injuries, metabolic syndrome, stroke. And all these pathologies are connected to an expressive oxidative stress and hypermetabolism of glucose in the brain. And in addition to that, multiple researchers have shown that oxidative stress and hypermetabolism not only connected to neurodegeneration, but also are early biomarkers of both acquired epilepsy and of the Alzheimer's disease. So they determine the development of these diseases. And with this positron emission tomography method, we showed the disorders in the utilization of glucose. And the cause of this hypermetabolism is unknown. However, it's interesting that hypermetabolism of the glucose of the brain among the patients with mild cognitive impairments without Alzheimer's disease definitely can forecast whether these cognitive disorders will progress and reach the Alzheimer's level or not. And in addition to that, multiple research on the epilepsy models among animals also show that the decreased glucose metabolism is something that can forecast and indicate the other severe stages. So the question is brain glucose hypermetabolism a consequence or a cause of disturbances leading to the progression of neuropathology? However, to answer this question, we've developed the model of chronic inhibition of glucoses. And we inhibit glycolysis to your glucose. And according to our estimations, this suppressed the glycolysis by not more than 14%. So here you can see the physical activity of hippocampus in this animal the course over 1 month. So daily, this glycolysis blockade was inhibited, and then the rate was indicated in red. You can see it on the slide. And the whole range of parameters that were characteristics of the epileptogenesis of hippocampus were also identified. And in addition to that, some animals were diagnosed with some spontaneous convulsions at the electrophysiological level. You can see it in the black here, and also at the behavioral level. So the histological analysis also identified the epilepsy-specific changes in the brain. So we observed the death of neurons in the field CA1 of hippocampus, and also the sprouting of the mossy fibers. over the dental gyrus. So this could be the trigger of epileptogenesis. Why glucose hypermetabolism initiates pathological process? Here, I believe that we need to say a couple of words about the importance of glucose metabolism in cells. It's wide known that as a result of glucose, of glycolysis, we have this fast ATP in mitochondria, the adenosine triphosphate, and this cannot be replaced by other metabolic matters. This is an exclusive one. And so here we can see the fast adenosine triphosphate involvement during an active brain activity. Also, the glycogen is there, and this is the only source of energy in the brain. And also the efficient antioxidant defense, which should prevent the oxidative stress. Oxidative stress is a very powerful regulator when it comes to the aging and different neurological disorders. And normally, the active forms of kislorod, ROS, you can see it here on the slide in the violet. And usually we get it from a couple of sources in our brain. Most of them are subproducts of metabolic reactions, and they are offset by antioxidant systems in the cells. Normally, the cells sustain the balance between the product of ROS and their neutralizers. However, if there is any pathology, the ROS products can prevail over its neutralizers, and as a result, this could incur some stress and multiple damage of the cells. And so we expected, obviously, that antioxidants could be efficient in fighting the oxidative stress in our brain. And as a result, they could also be active while treating different neurological pathologies. However, multiple clinical trials with the use of antioxidants did not confirm their efficiency. And so we've come up with the question, why? It's logical. And when we started to see what's going on there, we've seen that there is no data in the medical works connected to the antioxidants and the oxidative stress. And to fill this gap, we used these methods to register extra-cell peroxide in slices of hippocampus. And this allowed us to observe dynamically the release of active oxygen forms from the inactive cells. And we've shown that antioxidant tempole, you can see it over here at the bottom, in the up of the slide. And if it's concentrated highly, it reduces the release of the peroxide during the synoptic stimulation only by 20%. So these gray columns over here, this is about the time of the electrical stimulation on the referent fibers. The black line here is the control release of the peroxide, and the green line here is at the backdrop of this antioxidant. So we checked another antioxidant, apocynin, and it decreased the release of the peroxide by 50%, approximately. But what's most importantly here, when we provoked convulsions in slices, this antioxidant did not influence the release of the ROS at all. As you can see over here on this graph, the spontaneous convulsions started from a very powerful, fast release of a peroxide. And this result explains the inefficient treatment by antioxidants during clinical trials. Apparently, the therapy should be focusing not at the deleting the ROSs, but at the cause and the source of their production. So, as I've already said, most of the ROS are the byproducts of different biochemical reactions. However, there is one molecule which is known, this one, and its main function is the direct production of the ROS. This is the NADF oxidase, or we also call it NOx. And the most active one is NOx2. It consists of 7 units. They're localized in the cell membrane, and it also has a couple of subunits in the cytoplasm. And in response to the activating signals, those units are aggregated in one big molecule, and they start to produce the active forms of oxygen, and they transmit the electrons from NADF to the oxygen. And the trials have shown that during a pathology, NOx2 actually stresses the cells a lot. And this molecule can be a pharmacological target for direct and efficient therapy. So, can NOx be a pharmacological target for direct and effective therapy? For this, we conducted some research based on the epilepsy models and Alzheimer's models. Our experimental methodology included electrophysiology in slices of the brain and electrophysiology in vivo in animals while they were under anesthesia and while they were just behaving freely. And we observed the changes in metabolites, such as changes in glucose. You can see it over here, represented by the red line, lactate, green, glutamate, and oxygen. And in slices, in our physiological matter solution, we have 5 mMs of glucose. In the stimulation of nerve collaterals, you can see it over here, it's indicated in the green column, results in a massive activation of neurons with intensive consumption of oxygen and release of a lactate. And that's why on the graph in here, we can see the decrease of out-cell concentration of glucose and oxygen, and we see the increase in the level of lactate. In vivo, the situation is more complex since the activation of neurons provokes the release of substrates, submatters from arteries, and they are higher than the demand from the cells. Over here, you can see that after the synaptic stimulation, despite the high consumption by the cells, the out-cell level is rising due to the fact that it's coming from the blood. And afterwards, I'll represent you the results on the epilepsy model and then on the Alzheimer's model. In this slide, we can see epileptiform activity in slices of the brain. It was induced by epileptogene 4AP. The electrical activity is represented by the green color, and in parallel, we registered the concentration of the peroxide in the intercell space, and it is also represented in the green here. So, an epileptogene provoked very intensive interactile activity, and this interactile strike was accompanied by an emission of a peroxide. Over here, you can see the induced seizure that has been provoked by an electrical stimulation. The beginning of the seizure is accompanied by an emission and release of a peroxide, and it's 10 times more than during the interactile activity. And so this fast release of the peroxide during the interactile activities was fully prevented by non-selective antagonist, NOx antagonist, celestrol. It was also controlling the release of the peroxide during a seizure, induced seizure. And what's more interesting, this inhibition of NOx resulted in full prevention of any seizures. So we can see none in here. So the activation of NOx is the trigger of the seizure activity. We also studied separately some delayed effects of this stress. And it turned out that after the full removal of 4-AP and after the stop of all seizures, still the consumption of glucose is still reduced. So after the seizure, you can see the consumption of glucose indicated in red. So we have a full cycle. Not only the hypermetabolism of glucose induces the oxidative stress, but it also works vice versa. We checked our results against the in vivo method. We have anesthetized mouses. Epileptic gene provoked these powerful seizures, and they fully were blocked by selective inhibitors NOx-GSK. And in addition to that, we also tested against other NOx inhibitors, celestrol and others, and we got the same results. They reduced the number of seizures significantly. You can see it over here in this graph, as well as the duration of a seizure. That's how we managed to demonstrate that NOx activation is the trigger mechanism of seizure and the glucose hypermetabolism. And the NOx blockade could be an efficient method in target therapy treating epilepsy. Now I'd like to pass on to the Alzheimer's model. As I said, the early stages of the Alzheimer's disease are connected to the glucose metabolism and oxidative stress. And in a slice of hippocampus, we've shown that amyloid beta reduces brain glucose utilization via activation of NOx2. And it's quite known that amyloid beta is the direct activator of NOx. So we checked whether this selective inhibitor of NOx could prevent hypermetabolism of glucose. And it turned out that NOx inhibitor, you can see it over here in this graph, it's in blue. NOx inhibitor fully prevents the toxic effect of amyloid beta. Our results clearly demonstrate that NOx activation induced by amyloid beta is the main trigger of glucose hypermetabolism. To check our results in vivo, we injected the beta amyloids in mice in their brain ventricles. And here you can see the glucose level in black in response to the synaptic stimulation. Those vertical lines are the synaptic electrical stimulation. And as was expected, the out-cell level of glucose increased after an injection of amyloid beta. And it indicated the reduced consumption by the cells. However, while amyloid beta was injected against the backdrop of a NOx blocker, the glucose response and rates were at the control level, and so they indicated the full prevention of the toxic effect. That's how our data show that amyloid beta and NOx activation could be behind the glucose hypermetabolism. And it's known also that the hyperactivity could be registered at the very first and initial stages of the Alzheimer's disease. And when we inject the the beta amyloid into brain verticals of mice, we can actually see what's happening. It's demonstrated in the graph. And over here, you can see the graph of the integral activity, and it demonstrates the overall level of the neural net activity in dynamics. The next question is whether the oxidative stress is responsible for neuron activity, hyperactivity, during the Alzheimer's disease. So we injected amyloid beta at the backdrop of blocking NOx. And it turned out that blocking NOx prevents the development of hyperactivity in neuron tissues that were provoked by amyloid beta. And here you can see the average graphs of the activity 1 hour after and 48 hours after. Control is 100%. And one can see that increased electrical activity, it's over here in red, because of the amyloid beta, persists even after 48 hours. That's how we conclude that NOx activation by amyloid beta apparently also lays as the basis of hyperactivity of the network. So oxidative stress could be a general trigger for glucose hypermetabolism and for the hyperactivity. It's known that the Alzheimer's disease starts 20 years before the people actually are diagnosed with that. And in addition to amyloidosis, there are different biomarkers, and those are neuro symptoms such as apathy, depression, aggression, and anxiety. And we pay attention to the fact that our mice become quite aggressive after introducing these matters. And we wanted to understand whether NOx2 blockade could also affect the aggressive state of these mice. And for that, we introduced cannula in mice, and we separated mice to offset their social roles. We used two tests. So we have special obstacles as well. And here you can see the timeline of the tests. 1 week after the injection of amyloid beta and 2 weeks after that. So the mice were put in this compartment, and we recorded the number of fighting and some aggressive behaviors. And in red, you can see the group of the mice with the amyloid beta. In this group, the aggression level increased quite significantly and drastically. However, when beta amyloid was injected against the backdrop of NOx2 blockade, in that group there were no fighting at all. So the mice were quite active but not aggressive. And the second test that we conducted, those are the tests with obstacles. And it resulted in the same conclusions. Here we have 2 compartments, units, and it has a special obstacle with some holes. There is 1 mice in each of them, and the more aggressive the mice, the more contact it has with the obstacle. So it tries to establish contact with its partner across the other compartment. And with just amyloid beta, we've seen that the number of contacts with the obstacle and the duration of this contact was quite high. So it was quite different from the control ones. But when we had NOx2 blockade and amyloid beta, the indicators were very close to the control ones. And that's how we indicated that we could prevent such symptom as aggressiveness with such medication and treatment. And let me introduce the conclusions. So the oxidative stress associated with glucose hypermetabolism are likely the primary triggers of Alzheimer's disease and epilepsy. Then brain glucose hypermetabolism is induced by NOx activation. And third, inhibition of NOx may be the efficient way to prevent Alzheimer's disease and acquired epilepsy. And by this, I thank you for your attention.